All right, if you remember, I was telling you about how when Mendel did his experiments, he basically concluded by counting the number of seeds and plants he had of different types that it seemed like there were two genes or alleles or traits that were inherited from the parents for each plant. And each plant had two copies, and one copy could be dominant and the other could be recessive. And the dominant one would override the expression of the recessive one, but the dominant one didn't actually change the recessive one. The information was still there, so that if you crossed them, if you crossed your F1s, that in your F2s you could come out and see those same qualities again. You could see that recessive trait. He also figured out that if you're looking at, for example, smooth seeds versus wrinkled seeds, that the smooth versus wrinkled and the yellow versus green were independent of one another, meaning that the smooth trait didn't have anything to do with the color being yellow or green. Okay, so that led him to two of Mendel's laws, more mathematic laws, really more than anything. Two laws he came up with: law of segregation, which basically is that organisms have two copies of each gene, and that they segregate or come apart during gamete formation. And remember, gametes are sex cells. So basically, like for eye color, let's just say you have, although this isn't entirely true, it turns out it's more complex than this, but let's say you have a gene for brown eyes and you have a gene for green eyes. You have two alleles, okay? You have one brown, one blue. I don't know if I said blue or green, but let's say brown and blue. If you have one copy of brown and one copy of blue, you're going to have brown eyes, because brown, it turns out, tends to be dominant over the blue-eyed color gene. And it turns out there's a whole bunch of other genes involved with eye color, so it isn't quite as simple as that. But that when your parents go, or you go to make a sperm or an egg, the brown is on one, say, chromosome 4 or 5. The other chromosome will separate independent of that one. So that's the law of segregation, that those two copies that you have will separate. And then the law of independent assortment is essentially that different genes will separate independently of one another. Once again, if you're smooth and wrinkled, or if you're smooth, if you're a smooth gene, or you have a smooth gene, or you have a wrinkled seed, that has no impact on whether you're going to inherit the yellow or green color. They're independent and assort independent of one another. So that's basically the formation of Mendel's laws. But it turns out there were a lot of things Mendel couldn't explain that we now know in more modern genetic times, that the genetics that Mendel did, although groundbreaking at the time, very interesting, and gave us a tremendous amount of knowledge about genes, it turns out it's not always quite as simple as that. And there's many kinds of variations. So we'll go through some of the variations of things that we see. One of them is called incomplete dominance. And what happens in incomplete dominance is it turns out you have two genes, say red and white. Okay, so snapdragons, another different type of flower, have similar kind of a similar flower as the pea plant, but we have this flower here called a snapdragon, and we have a gene that gives it a red color, but we also have a gene that gives it a white color. So that's all pretty normal. But then it turns out if you cross the P generation, the pure breeding or parent generations of reds and white, what you get instead is you get in the F1, you get pink. You get all these pink ones. So you get, it turns out that red is, although it is the dominant color, it turns out it's not quite fully dominant, and so you get a sort of an in-between effect. If you cross the F1s, the pink ones, what you get is you get a 1 to 2 to 1 ratio in the F2, which is you get one red for every 2 pink for every 1 white. Now you'll notice over here what they did, the way they did this was they put a C to represent the gene, and then the R represents the allele in that particular gene in that case. Okay, so this is another way you can show genetic differences. This is basically the same idea as having the letter, the R or the W, but here they put the C to let you know they're still talking about flower color or whatever the trait might be. The letter is actually irrelevant, and then the R is supposed to represent the red flower, and the W is supposed to represent the white flower. Could also use a lowercase r if you wanted to do that in next to that C. So that gives you all pink because all of them in the F1 end up with one copy of the red, one copy with the white. And then in the F2 you get what you see down here. All right. Mendel also, the other things that Mendel can explain, another one found in blood type, is actually two of them here, multiple allele systems and codominance. Okay, so the idea with multiple allele systems is it turns out, in Mendel's case, he had yellow seeds or he had green seeds, or he had wrinkled seeds or he had smooth seeds. But it turns out for some things there's more, even though you only get two copies, one from mom and one from dad. The variation of copies of alleles that are out there can be widely ranging. So for example, blood type is an example of a multiple allele system in which there is a blood type for A, and there's a blood type for B, and there's a blood type for O. So there's three different alleles in this case, just for the A, the B, or the O. Okay? It's also an example of codominance, and that's where if you have one copy and you have the other copy, instead of one being expressed and the other not being expressed, they're actually both expressed at the same time. And that's how you get blood type AB. So it turns out that something like blood type is not only is it a multiple allele system, but it's also an example of codominance. So you can have one copy of the A in your blood type A. You can have one copy of the A and one copy of the B like you see down here, and then your blood type AB. A and B are both dominant over O, which is recessive. So often with blood type they use this I, and the capital I represents the dominant trait and the lowercase i's represent the recessive trait. Okay, so that's in a multiple allele system and codominance. And then you might be wondering about, well, what about the positive and the negative? That's called the RH factor, and that is a simple dominant recessive. If you are RH positive, that means you have at least one copy of the RH gene, allele, and that produces a particular structure that's on your cell type, just like the A's and the B's do. Okay. Other thing Mendel couldn't explain or would have trouble with is this idea of pleiotropy, where you have one particular gene that alters many different phenotypes. So sometimes a gene doesn't just make a flower yellow. It might be doing a whole bunch of things. An example of pleiotropy then would be in humans there's a genetic disorder that people can get called sickle cell anemia, and it's a genetically inherited blood disorder in which basically you make red blood cells incorrectly. This shows you normal red blood cells, and these show you some that have been altered or they're sickled and damaged because of this one protein called hemoglobin that isn't made correctly. Well, because of that one gene, you see several different phenotypic issues. So a person might have poor kidney function, they might have pain and poor blood circulation in their hands and in their feet. They might have issues with eyesight, basically all coming back to the fact that these red blood cells are made incorrectly. They could be at higher risk for strokes and things like that. Once again, all being related to this one thing, but phenotypically you might see several different problems in one person phenotype with this phenotype because of this one gene. So that's pleiotropy, where one gene affects many different phenotypes. Epistasis is another example, and in epistasis what you have is where one gene that will alter the expression of another gene. Okay, so you can have a dominant recessive gene, but whether that dominant recessive gene even operates or works could be dependent on the function of another gene. So what you have up here, this is an example showing you a bunch of mice under an example of this epistasis. And so what happens is there's this C allele. Okay, so there's this C allele, and you have to have the C allele to have pigment be deposited into the fur of the mouse. Okay, so what ends up happening is the fur color then is under the effect of whether you have big B. So big B gives you sort of a dark colored mouse. Okay, little b will give you a white colored mouse. But the problem is you have to have, notice this one here, for example, this is little b, but because it has a copy of that big C, okay, it has a copy of that big C, this mouse looks different from this one because you end up getting some pigment deposited in the fur of the mouse anyway. So in this case, the C allele will alter or affect what the other one does, the B gene or B allele in that case. Okay. Another one is polygenic inheritance. Simple idea here is that sometimes a particular trait is actually coded for by many different genes. So people's height, your height, for example, or your weight, or your hair color. Actually, many human characteristics that you think about are an example of polygenic inheritance, which means you have a whole bunch of genes that sort of work together to give you a particular phenotype. So you know, if your parents are tall, then there's a good chance you're going to inherit many of those genes, and you're going to be tall, but that's not always the case because there can be a variety of factors of genes that are at play there. And then the last one is this idea of linked genes. And what happens in linked genes is, we talked about Mendel's law of independent assortment, that basically, like if you were wrinkled, as far as the seed goes, if you had a wrinkled seed, or you had a smooth seed, that had no impact on whether you were yellow or green. But it turns out that sometimes that can have an impact. Meaning, like, for example, this is a very well-studied animal here, this Drosophila, this fruit fly, and it turns out there are certain genes and certain characteristics they have that can be located on the same chromosome. So, for example, a black body and this cinnabar-colored eyes, okay? It turns out that those are on the same chromosome. And what happens is, if you inherit the black body type, you have a higher chance then of getting this gene with it, because they're what we call linked genes, because they're on the same chromosome. Now, let me remind you though, if you think back to meiosis, remember that crossing over occurs where chromosomes, where homologous chromosomes pair up and they swap places. So you don't necessarily always get these two together because crossover can occur. So it turns out you can measure or map. You can make these maps of how much crossover you get or how much they independently assort, and based on that you can figure out where they are on a chromosome and about how far apart they are. So that's the idea of linked genes, that if you get one, you have a higher rate of getting the other, versus Mendel's law that it was just separate and random.